Hello and welcome to another episode where we'll be discussing the MCQs that were given previously and we'll try to explain it. Okay, so if you don't follow the page already, go check out the Instagram page where all these MCQs are given with explanations. So gastric lipase is secreted by parietal cell, chief cell, mucous neck cell, enterochromaffin-like cell. So the answer here is your chief cell. So we are talking about lipase. So talking about lipid digestion, it basically starts in your mouth, and as we go down, it goes into your stomach and then it continues in your intestine. The major role is played by the intestine, but a minor role is played by these other things. It starts off in the von Ebner's gland. It's not actually your salivary secretions, but in fact secretions from your von Ebner's gland that secretes your salivary lipase. And then in your stomach, it is your chief cells that secrete your gastric lipase. If you remember, chief cells also secrete your pepsinogen. So chief cells basically secrete pepsinogen as well as gastric lipase. The other cell which is there, which is a parietal cell, is responsible for acid secretion, and it is also helpful in secretion of intrinsic factor. So parietal cell is for your intrinsic factor as well as acid secretion, and chief cell is for pepsinogen secretion and gastric lipase. So this is basically what initially happens. Okay, and most fat digestion begins in the duodenum. So in the duodenum, we have your pancreatic secretions which are coming, which is pancreatic lipase, which is the most important one, which acts on the triglyceride, and then co-lipase, which actually helps act pancreatic lipase. Okay, so this co-lipase actually binds to the fat droplets and helps attach the pancreatic lipase onto the whole thing. Okay, so that makes a mixture. And you have your cholesterol ester hydrolase. So that is for cholesterol esters. Okay, and your phospholipase A2, that is for the phospholipids. So these help digestion of lipid, and this is the major thing. So we had asked this question last time. In Caisson's disease, why is the majority of effects due to nitrogen and not the other gases? So to answer this, what you have to understand is in your atmosphere, 78% of the composition is basically your nitrogen. Nitrogen is relatively inert. So compared to the next ones, the oxygen and the carbon dioxide, this is relatively more inert. It's not actually an inert gas. That's why it forms nitrous oxide and all that. But it is comparatively more relatively more inert. Okay, especially in our body. On the other hand, oxygen, carbon dioxide and all gets utilized, metabolized, forms other products, byproducts and all that, and it gets removed in different ways. Not your nitrogen. Okay, so that is why your nitrogen is playing a major role, because one, it is more in composition, two, it is relatively more inert, so it's not used up. So its composition and its effect is going to be more pronounced, especially during a sudden ascent. So that is why you have to remember the difference. Deep sea diving, staying under at high pressures for a long time, that is one thing. In Caisson's disease, it's not basically that. From that compression, okay, from that pressurized compressed environment, we are all of a sudden ascending suddenly. That is your Caisson's disease. Okay, this is where some students find it difficult to differentiate those two. Okay, staying underground, deep sea diving for a long time, that is a completely different effect. But from a pressurized environment, all of a sudden decompressing, that is your Caisson's disease. Okay, so let's just first understand what happens when we go deep down. So when we go deep down, the column of air that is above us keeps increasing, so that basically increases the pressure. The partial pressure of all these things keeps increasing. So nitrogen, oxygen, carbon dioxide, this is the major components, and their partial pressure keeps increasing as we go down. Okay, and because of that, at high partial pressure, basically these gases gets dissolved and solubilized in your water in your body as well as the fat. Okay, so we have your fat and we have the water. At high pressure, once the person goes down, it goes inside, and because they are continuing to remain in this high pressure, it's not going out. So this, at high pressure, it goes in and it's even if it wants to come out, because it's continuing to stay there, it's staying there. So the main problem here is your nitrogen narcosis, oxygen toxicity, carbon dioxide toxicity. These are the problems if you continue to stay deep down for a prolonged period of time. Okay, so next, the person ascends subsequently. Okay, so now when there is sudden ascent or decompression, the partial pressure has become normal all of a sudden. So this outside pressure which is there is no longer there. But inside it was solubilized at a very high pressure. So now what is going to happen when it suddenly ascended and the outside pressure is no longer there to keep it inside? It's suddenly going to start coming out. And when that happens, all of a sudden, it starts forming bubbles. Okay, and these bubbles are what majorly causes problems. So that is basically your Caisson's disease. Okay, the majority of the problems happen in your joints and the muscles. 85 to 90% of the problems are in your joints as well as the muscle. And then 5 to 10% of the symptoms are nervous, 2% and respiratory. Okay, so this is basically what happens with your decompression, sudden decompression. So also remember that Caisson's disease is not only for deep sea diving. Any pressurized environment where high partial pressure is there, you suddenly go out. Okay, this can even be there for pilots who are pressurized and all of a sudden that pressurization is gone. Okay, and it's low partial pressure outside, that is there. You go to, say, moon, Mars, whatever, you go outside. Okay, so there are certain procedures over there. Okay, when you man a vehicle outside, so there is differences in the pressure inside, outside. So any problems coming there, all of a sudden the pressure changes, again this problem is going to happen. Okay, so that is why over there you need to spend some time to adjust stuff. Okay, so you should not suddenly go on to change your partial pressure. Which of the following statements true about clathrin vesicular transport? Formation of clathrin cage requires ATP. No, it does not. Okay, it happens naturally. Dynamine uses ATP hydrolysis for budding. It uses GTP in fact. Uncoating of clathrin cage requires ATP. So that is your answer. The clathrin coated vesicles are trafficked to form caveosome. It's not caveosome, that is for caveolin mediated transport. Here it goes to your endocytic vesicles. So you can see over here, initially the formation of the clathrin coat, there's no ATP required for this. And after that, the coated vesicle which has come out, that requires ATP to uncoat and then further on go down. Okay, so this is the place where ATP is needed. And for this pinching using dynamine, that requires GTP hydrolysis. Okay. Example of rapidly adapting sensory receptor with smaller receptive field. So the answer here is your Meissner's corpuscle. Let's just try to discuss this. So when we talk about corpuscles, okay, so corpuscles basically are covered with fluid inside and a nerve terminal which is there inside. Okay, so these corpuscles are rapidly adapting. Okay, so sudden changes which are there, they detect it and then they adapt to it so that again small changes can be detected. So this is basically based on the fluid that is displaced. Okay, now apart from that we have the expanded tip, the Merkel's disc and the Ruffini's ending which form slow adapting. Okay, so Meissner's corpuscle and Pacinian corpuscle, the corpuscles are rapidly adapting while your Merkel's disc and Ruffini's ending are slow adapting. Now out of this, these, the Pacinian corpuscle and Ruffinian, which are much more deeper in your skin. Okay, so these have a larger receptive field. What it means is your one Pacinian corpuscle can detect a larger area. So whether you touch it here or here or here, it's all basically detected by one single Pacinian corpuscle. On the other hand, your Meissner's corpuscle and expanded tip receptor, which are much more superficial in nature, are for smaller receptive fields. So if you touch here, it's one Meissner's corpuscle. If you touch here, it's another Meissner's corpuscle. Okay, so the rapidly adapting and smaller receptive field, the answer is going to be your Meissner's corpuscle. So just remember this, and whatever permutation combination comes, you can just answer it. In the above diagram of leaflets of membrane, label A, B, C correctly. So there are a lot of confusing things here. Flippase, floppase, scramblase and all that. So the answer here is this. Okay. So let's try to understand this. There are two leaflets in your membrane. We have the outer leaflet and the inner leaflet, and the constituents of that vary a lot. Okay. So Flippase is something that transports some phospholipid from outside the outer leaflet, extracellular, to the cytosolic side, the inner. Okay, that is Flippase. Floppase, what it does is from the inner leaflet, it takes it outside, and both of these require ATP. On the other hand, we have your scramblase. So scramblase, what it does is it does both. Okay, it can move something from outside to inside and from inside to outside. Does not seem to need ATP, and uh, it you known to use something like calcium, for example. So not ATP related, but it's known to use calcium. Okay, and this whole mechanism is very important in your body because problems in this can result in a lot of diseases and all that. Okay, for example, a simple example, phosphatidyl uh, serine, which is there. Okay, so phosphatidylcholine is there outside, phosphatidylserine is usually inside, ethanolamine, serine and all are usually in the inner leaflet. By some mechanism, it goes outside. Okay, so it going outside means it's basically activating a lot of mechanisms that are related to autophagy, for example, or apoptosis, or it can even, inside in your blood, the whole coagulation cascade is activated. So these are various ways by which these are actually important to maintain a lot of things in the body and for some sort of signal transduction as well. Smaller units that make up gap junctions are. So the answer here is connexon. Small, even further is your connexon. So you can take a look at this over here. This is your gap junctions that you see over here. Okay, so this is one example of how an electrical synapse is going to be. So this has basically two channels are there on either side. Okay, so that is why on one side what is there is called the hemichannel. Okay, so two such things are there that forms the whole channel. Okay, so this is your gap junction, and in the gap junction, this one whole thing, this one hemi channel on one side is called the connexon. Okay, and this connexon is further made up of connexins. Okay, so one hemichannel, that is one connexon, is made up of 6 connexins. So one connexin is basically, you can see here, it is made up of your 4 looping channels. Okay, so membrane spanning segments, 4 of them are there. 1, 2, 3, 4. So this is one connexin. 6 such connexins form your one connexon, and that is the building block of basically your gap junction. 2 of those connexons on either side forms your gap junction, and that forms part of your electrical synapse. The most powerful buffer system in the body is, the answer here is your kidney. Okay, so when you talk about the buffer systems in the body, the first line, when something changes, the immediate thing that takes care of it is your chemical buffers in the body fluid. After that, second line is your respiratory system, and the third line, so the most delayed one is your kidney, yet it is the most powerful one. So when you talk about chemical buffers in more specificity, the bicarbonate buffer system is the most important one. So in the chemical buffer system, if the same question was asked, the answer would be bicarbonate buffer system. Okay, and then there are non-bicarbonate buffer systems as well. For example, phosphate buffer system, proteins. Okay, so phosphate is more important intracellularly, and it plays a role in your kidneys as well. Your proteins, for example, because of their zwitterion nature, they also play a role in buffering. Okay, so one major example of that is your hemoglobin structure. So these are the different chemical buffers in the body, and the most important one is your kidney. Migratory motor complex occurs at intervals of, so the answer here is 90. So of the lot, this is the best answer. So if you take a look at it, when you talk about migratory motor complex, this happens at the time of fasting. Okay, so interdigestive period is where migratory motor complex happens. After feeding, in the fed state, your basal electrical rhythm happens. So please understand the difference between those. So when you talk about migratory motor complex, normally majority of it is a quiescent phase, not much activity at all. Okay, that is the initial phase, phase 1. And in phase 2, irregular bursts of activity happens. It's not a continuous one. Irregular bursts in between happens. This is phase 2. And just as the phase 2 is ending, from the M cells in the duodenal mucosa, something called motilin is released. So this is just one reason. Okay, there are a lot of other reasons. At your level, you just need to understand this. Okay, so your M cell plays a major role here, and that starts your phase 3. Okay, so now it's not irregular, a regular burst of activity is there. Okay, so again it goes to the quiescent phase. Okay, and irregular bursts, regular bursts. Okay, so from when this started to when the next one starts, this interval is 90 to 120 minutes. Okay, so this is the answer that was asked. Okay, so this is 90 to 120 minutes, and each of this phase that starts, okay, basically it starts in your stomach. Majority of it starts in the stomach. 75% starts in the stomach. On the other hand, 25% is said to start in the duodenum and proximal jejunum. Okay, and later on it ends. So this phase 3 that we're talking about usually lasts, so we say, from 5 to 10 minutes, on an average 7 minutes, and can go up to the amplitude of, on an average, 30 millimeter. Not up to, an average of 30 millimeter mercury.